All right, so here we go. Next, next intro video. I'm sure you've all been waiting for this with bated breath. So we're going to be talking some more about PCR today. Um, I know that a lot of you still haven't watched this video, the video I call the best PCR video. So make sure to go check that out. It's pretty great. So we've been talking about how to analyze life at the DNA level for the most part so far. Um, and today we're going to move over into the next level, so at the RNA level. So whenever we're talking about things that aren't the DNA itself, we're typically talking about gene expression. Okay, so the DNA is part of the DNA is genes. And in order for the information that's in DNA to be used, it needs to be expressed. So anything coming off of the DNA in order to be functional is going to be considered gene expression. And generally, the first way we look at gene expression is at the RNA level. So just to remind you of this structure, our DNA is in that double helix anti-parallel. As it's transcribed, the RNA is going to be in the 5' to 3' direction. So it's actually reading the 3' strand of the DNA. So again, I'm going to mention this later, but the RNA is always going to be in the 5' to 3' orientation when we're working with it. And that's what life winds up using. So it has to read, the transcription has to be read off of a 3' strand. So how does transcription work? And it's way more complicated than this, but this is a pretty good overview, and it's more than what we need for the purposes of this class. Um, so transcription starts with a region in the DNA called a promoter. And there are multiple elements of promoters, and every promoter is pretty different, but there are little elements within the promoter. Um, and these are recognized by proteins called transcription factors. Okay, so the promoters are DNA elements that are present in the DNA that are recognized by proteins called transcription factors. And so these transcription factors bind the DNA, and then they recruit a protein called RNA polymerase. In the example we're going through is for creating messenger RNA, it's RNA polymerase 2 in eukaryotes that we're going to be that the cells use. So the transcription factors recruit RNA polymerase. The RNA polymerase it hops onto that 3' strand, and it starts making a 5' strand copy of the DNA as RNA. And so I forgot to include a specific slide on this, but the main difference between DNA and RNA is that, so if you remember our bases, our carbons in the ring structure here of the ribose. So DNA, um, it's considered deoxyribose because there's not an oxygen here. So again, this is our 1, 2, 3, 4, 5' carbons. And so here at our 2' carbon in RNA, there's a hydroxyl group, an OH group is sticking on this 2' carbon. In DNA, there is none. We only have the 3' OH group. Also, instead of the base thymine, we have the base uracil in RNA. Okay, so again, promoter in the DNA gets recognized by transcription factors, which recruit RNA polymerase 2. RNA polymerase 2 makes an RNA copy of DNA. And this is kind of particular to eukaryotes to a certain extent, but as the RNA is being transcribed, okay, so the RNA polymerase 2 is the enzyme that actually does the transcription. Um, a enzyme comes in and creates a 5' cap on the messenger RNA. So it signals that this is the beginning of a messenger RNA sequence. Um, Ultimately, the RNA polymerase 2 is going to transcribe a big chunk of DNA into RNA. Eventually, it's going to finish process called termination. And at the end of the messenger RNA, we get something at the 3' end of the messenger RNA, we get something called a poly-A tail, basically just a string of A's that winds up getting put onto our messenger RNA. Then a process called splicing comes in. Um, and so here's an example of a eukaryotic gene. There are introns found in prokaryotes, but they are pretty rare. This is a much more common eukaryotic phenomenon. So if we're looking at this gene structure, and so there's a website we're going to use next week called the NCBI Gene Database, we can see the structures of genes. Okay. And so this right here is, it's telling you, within our chromosome, this is our landmark. It's basically mile markers in kilobase pairs. Um, and so this is our gene structure. Up here would be the promoter. And what this shows you is the messenger RNA, or the RNA that's transcribed off of that gene. And so that's shown in green here. And so the promoter would be in the DNA, and then the RNA gets transcribed off of that. And so this whole big length of thing, which is around 45,000 base pairs, is considered the pre-messenger RNA. This whole region gets transcribed off of the genome. But in messenger RNA processing or splicing, there are regions of the RNA that get cut out. And those regions that are cut out are called the introns. And the regions that stay in the messenger RNA are considered the exons. Okay, and so in this diagram, these thin lines that connect these vertical bars, the thin lines are the introns, and the vertical bars are the exons. So all these thin parts with the arrows in them get cut out, and these other parts stay in. And so you can see in this particular example, our pre-messenger RNA is 45,000 bases, but by the time splicing gets done, we're down to 4,390 nucleotides. When we're talking about a messenger RNA sequence, we should refer to it as NT, or nucleotides, because messenger RNAs are linear molecules. They're not base paired necessarily. And so we refer to it as nucleotides instead of base pairs. When we talk about DNA, we're typically going to be talking about it being base paired to another thing. So we refer to it as base pairs. With this one, you'll notice that there are two individual pieces of RNA that are transcribed from the gene SLC16A1. And what this represents, there's two slightly different promoters. And so really this promoter would be a little bit more up here. This promoter would be a little bit more back here. But ultimately, we'll talk more about what gets encoded off the protein or off the messenger RNA next week. But ultimately, these two wind up making the same protein. Okay, so at the beginning, so this is another way to represent messenger RNA splicing. And so people draw these boxes, and then these lines that are these V-shaped lines represent the introns. The bigger boxes represent the exons. Again, at the beginning of the messenger RNA, we would typically have a cap put on there. It's a modified G base put on backwards. And then we have a little bit of messenger RNA that's not encoded into protein. Then we have what's called the protein coding region, or also known as an open reading frame. And then we have a little bit, another region of the messenger RNA that's not translated in the protein. We call this the 3' untranslated region. This part that's not translated in the protein is called the 5' untranslated region. Again, messenger RNAs always start at the 5' end, go down to 3' end. And down here at the 3' end, we have our poly-A tail. And so this diagram is showing three A's on there. In reality, this can be hundreds of A's in length. Okay. So in the grand scheme of things, first the cap is added to the 5' end, then the poly A tail is added, then the introns are cut out and the exons are ligated back together or spliced back together. The introns are spliced out, and in that process, the exons are ligated back together. So again, just to remind you, we're always working with 5' to 3' direction in the messenger RNA. But when we're working with messenger RNA, we can't, or any RNA, we can't PCR directly off of RNA. Okay. And so DNA polymerase cannot use RNA as a template, right? So when we were doing PCRs last week, you remember our template was our plasmid DNA, right? This week, our template is essentially RNA, but DNA polymerase or attack polymerase cannot, any kind of DNA polymerase really cannot amplify directly off of messenger RNA, right? So if we try to get all the messenger RNAs out of cells and do an RNA extraction, we can't use that as a template for PCR. Okay, so what do? Well, we have this messenger RNA. It's got a cap. It's got a poly A tail. What we can do is take viral reverse transcriptase, and so there are a few different types of reverse transcriptases that have been utilized commercially. We can add a primer, right? Because reverse transcriptase is technically a DNA polymerase, and it requires a primer as a place to start transcription. And so if we add the primer as a string of T's, those T's are going to base pair with A, and we call this oligo DT. So it just means oligo means a short sequence of nucleotides, or a short sequence of anything, but in this context, it's a short sequence of nucleotides. So it's a deoxy T. Okay, so it's a DNA oligonucleotide, so 12 to 18 T bases. Those will base pair to the poly A tail that's on the messenger RNA. The reverse transcriptase will hop on to that primer and make a DNA copy of the RNA. Okay, and so this is a single-stranded DNA, but that's fine for our PCR purposes, right? If we have a primer that can anneal to that, it's going to copy and it's going to make a double-stranded piece of DNA in our first round of the PCR. So what we have here is we've now created a DNA copy of our RNA. And what this allows us to do is determine gene expression, right? If we go back to our DNA, say that we wanted to analyze gene expression from different tissues. So this week, you're going to be analyzing gene expression from brain versus kidney. Okay. If we extracted all the DNA from brain and all the DNA from kidney, that DNA would be identical. Okay. The DNA in all 30 trillion cells across human cells is identical for the most part, not including lens cells and certain types of immune cells, but that's a story for another class. But if we were to take all the brain cells and all the kidney cells, that DNA would be identical. And so analyzing DNA is not going to tell us anything about gene expression. We want to know what gets expressed off of the DNA, and we want to analyze how many copies of the RNA are essentially there. And so we use this cDNA as an estimate for the RNA that's present in a certain cell extract. Okay, so what we're going to be doing is PCR for particular genes of interest. Everyone's going to be doing PCRs for Amy's project. And essentially what we should theoretically see is that if we take some cDNA from brain and some cDNA from kidney, that we'll probably see expression of these genes in the brain, but not in the kidney. One of the things we should do in all of our our PCRs is do what's called a technical negative control, right? And so if you think about what we put into a PCR reaction, we have our TAC, we have our DNTPs, we have our primers, we have water, we have our buffer. In our technical negative control, we don't add any template. Okay, so the template here would be cDNA, right? cDNA is the DNA copy of the RNA that we've made. We have cDNA from brain. Here we have cDNA from kidney. And here we're just not adding any cDNA. Okay, so if we don't see anything here, that's a good thing. But if we don't see something here, that could mean a few different things, right? It could mean we just didn't assemble our PCR reaction properly. It could mean that the cDNA is bad. And so what we have to do is what we call like a loading control. And so we also have to PCR for what's called a normalizing gene or a housekeeping gene. And so we generally use something called tubulin, or we can use actin, or some people use GAPDH, which is, or G3PDH, glyceraldehyde 3-phosphate dehydrogenase. Generally, any kind of housekeeping gene is going to be something that's expressed in a large array of cell types. And so for our purposes, tubulin tends to work pretty well. It's expressed in most tissues, most cells. And so what we can say when we look at this is, okay, our cDNA reactions we know are good here. Our loading is similar between kidney and brain. So that means that our what we see here is that this gene really is only expressed in brain and not in kidney, and that there's not some problem with our kidney cDNA. Okay, so you're going to be setting up PCRs separately for a gene of interest and tubulin. And you're going to be having 3 reactions per set of genes. Okay, so you're going to set up what's called a master mix. We use master mixes for the sake of consistency across our reactions, right? If this is our reaction of, we're going to have our primer, our template cDNA, our buffers, our DNTPs, our TAC, and some water. We're going to multiply all these numbers times whatever. I have it shown up as times 6.5 reactions here. I always do an extra half reaction, and this accounts for pipetting error. I have this shown as 6.5. You're really only going to be doing 3.5. Okay, so you're going to be setting up master mixes of 3.5 times whatever goes in your original reaction. You put those all into a tube, then you transfer, you mix that all up real nice. Then you wind up putting whatever, if it's 24.5 microliters of this master mix into individual PCR tubes, right? Because this is all going to be the same. And then the template cDNA, your cDNA from kidney, brain, or no cDNA, is what gets added in last to the PCR tubes. Okay, so you're going to mix one master mix for your gene of interest, one master mix for your tubulin, and then make sure to mix these up really well, aliquot them into 3 PCR tubes, and then add your cDNA to that. All right, so you're going to do this. Again, it's going to be kidney and brain cDNAs. Then you're going to have 2 reactions, one for tubulin, one for your gene of interest. These primers are for genes that Amy is interested in. And so the primers are going to be in the methods 21 box. Just pick a random one and use that for your random primer pair and use that for your reactions. Okay, so you're going to set up your reactions. Again, it's about ½ hour to set up, 2 hours to run the PCR. And then you're also going to do 1.8% agarose gel of your PCR products. Again, this takes maybe a ½ hour to set up and an hour or so to run. Make sure to take a picture of your gel, label it, and post it to Moodle. One thing that's different, make sure you're doing a 1.8% gel. These are really small DNA fragments that we're going to be looking at this week. And so you want a higher agarose percentage in order to account for those smaller PCR product. So think about the difference between the amount of agarose that you added for your 1.5% gels that you've already done versus the 1.8% gel that you need to do this week. All right. And as always, feel free to hit me up with any questions that you have.